These doctors and researchers turned monsters and produced the world's deadliest bacteria, viruses, and chemical weapons and tested them on thousands of Chinese peasants, Koreans, Russians, and other prisoners of war. This is not a pretty story. Viewers with sensitive dispositions are advised to journey with us for the next few minutes with caution. But it is a necessary story so that the world would understand what happens when scientists and doctors play God. This story will focus only on the most brutal human experiments of Unit 731, a covert biological and chemical warfare research and development unit of the Imperial Japanese Army during World War II, headed by the infamous Surgeon General Shiro Ishii, a medical doctor and microbiologist. If you can watch through the third experiment, you are one of a kind. The first human experiment, testing of cholera vaccine. Dr. Ishii and his henchmen produced 2 cholera vaccines, one using an ultrasonic method and the other using a conventional method, a crude way of making vaccines. They forced 20 Chinese prisoners from their cells to the laboratories to test their products. 8 of these prisoners were given vaccines made with ultrasonic equipment. 8 others were infected with the cholera vaccine produced by the conventional method. The remaining 4 were not inoculated and served as control. These unfortunate victims were well fed with the healthiest foods available at the time. This was not an act of kindness or love. The purpose was to keep them in good health to yield useful data when they were subjected to test. After 20 days, all the victims were forced to drink large quantities of milk contaminated with cholera pathogens. The 4 who had not been inoculated became sick. The doctors observed them closely, recording all the symptoms, but provided no treatment. Shortly, these 4 died and their bodies were dissected and the internal organs were removed and studied as if they were some laboratory rats. One might say these 4 were lucky to die before dissection, as many victims of Unit 731 were dissected while alive. In some cases, vivisections were carried out on victims without anesthesia. With their bodies strapped to the table, eyes wide open, and mouths gagged with towels, these doctors from hell would cut open their victims as if watermelons. All 8 who got the conventional vaccine contracted cholera and died. Only those with ultrasonic vaccines showed no symptoms of cholera. But they too didn't escape the dissection table. In Unit 731, many victims who survived an experiment were dissected and their internal organs were studied. Generally, if victims survived an experiment and they were in good shape, they would be preserved and used for further experiments. But if feeble, they would be killed. A colonel killed 2 men after concluding an experiment and remarked, "The materials were no longer worth keeping for further work." 2. High-pressure test. A special pressure chamber was constructed to help the researchers determine how much pressure the human body could absorb. The test subjects were forced into these gruesome chambers and all kinds of measuring devices were strapped onto their bodies. Pressure was introduced and gradually increased until the victims collapsed in convulsion and died. This was a slow and agonizing death. As the pressure increased to very high levels, their eardrums ruptured, and their lungs overexpanded and collapsed. The high blood pressure forced their hearts to work harder, while gases in the midsection were compressed, causing severe abdominal pain. All these factors contributed to the victims' excruciating and slow deaths. Meanwhile, these doctors and researchers were busy with pens on paper, trying to discover the next breakthrough in medical science that would save the world. One might ask, why would medical doctors who were trained and saddled with the responsibility of saving lives and spreading healing upon the earth engage in such barbaric acts? One of the reasons Dr. Ishii and his accomplices engaged freely and enthusiastically in biological weapons human experiments was because they believed their victims were inferior beings who were sacrificed for a higher purpose. They also believed the world would be a better place to live, without so many of these subhumans wasting the planet's limited resources. 3. Frostbite. This experiment was performed during the coldest part of winter in Manchuria, when the temperature could reach as low as -20 to 30 degrees Celsius. Victims were taken outside in below freezing temperatures or placed in specially designed cold rooms. They were tied with ropes or chains to prevent any movement or attempts to shield themselves from the cold, leaving their limbs fully exposed. Water was repeatedly poured on the bare arms and legs to accelerate the freezing process. At times, electric fans were used to speed up the process too. As the water gets colder, ice forms on the exposed limbs. These scientists, with hearts of steel forged from the fire of hell, would chip away the ice and continue to pour water. The victims initially felt intense cold, but by now they felt severe burning sensations as their tissue began to freeze. The researchers meticulously documented the stages of freezing, including changes in skin color, swelling, blisters formation, and tissue necrosis. Victims were kept in this state for hours. The researcher would strike the limb regularly with a club and observe the sound it produces. When the limb produced a sound like a wooden board, then the researcher would know that it had completely frozen. Sometimes as the researcher hit the frozen limbs, flesh and muscles would detach and fall off, leaving the bone. Bones also became so brittle that they were shattered from the blow of the club. An eyewitness recalled that a Russian woman used for the frostbite test gave birth, and her baby too was subjected to this horrible experiment. After inducing frostbite, some of the victims were subjected to various methods of rewarming to study the effects and potential treatments. These methods often involved further painful and harmful procedures, such as immersing the frozen limbs in hot water or using heat lamps. Years passed, and one former Unit 731 member recalled that the victims had no hands and feet. These experiments were part of broader efforts to improve the survival and performance of Japanese soldiers in cold environments during military campaigns. So, these scientists did this out of love for their troops on the battlefield. For plague test. In 1944, Ishii and his team carried out respiratory tract tests to help them design an effective method for dispersing plague germs. This is one of the most deadly bacteria known to humans. Historically, plague pandemics, such as the Black Death in the 14th century, have caused millions of deaths in Europe, Asia, and North Africa. In Europe alone, it killed about 25 to 30 million people, which was about one-third of the continent's population at the time. No doubt Ishii wanted to cause another Black Death, except this time, in the camp of his enemies, the Russians and the Americans. He had many kilograms of the pathogen and was now looking for a suitable way to infect his enemies in large numbers. 10 human subjects were tied to stakes in the ground, each stationed a few meters away from the next subjects. Test tubes filled with solutions of plague germs were scattered among the subjects on the field. The plague germs were bred from the hearts, lymph, and spleens of infected rats. The test tubes were set up and timed to burst at a specific time. When they did, the plague emulsion broke into tiny droplets and circulated in the air which the subjects inhaled. The experiment was deemed a failure because it didn't give the wicked scientists the result they desired. The next test involved using deadly gases. Human subjects, along with dogs, pigeons, rats, and monkeys, were placed together in a glass-walled chamber. The aim was to compare how different animals responded to various toxic chemicals. The human subjects were blindfolded and put into the glass chamber with the animals. Then different toxic gases were released into the chamber. First, Adamsite gas was released. The subjects immediately began to sneeze and cough and later vomited. They became disoriented and lost their coordination. Some cried and shouted amidst the unceasing cough. After Adamsite gas, they used other lethal gases such as lewisite gas, phosgene gas, and white phosphorus. This chemical weapon burns at extremely high temperatures and causes severe burns on human subjects. It attacked the respiratory system of the victims and many died a horrible death. 6. Using food and drinks as conduit for pathogens. Mass-producing highly pathogenic bacteria and viruses was just one step in their murderous experiments. The scientists at Unit 731 needed to find the most effective method of spreading the germs to their enemies. To achieve this, they set up an experiment using different types of food as vectors for the pathogens. Chocolates were filled with anthrax bacteria, and cookies were laced with plague bacteria. Drinks were also contaminated. Tea, coffee, milk, water, beer, and whiskey were all infused with specific quantities of pathogens. Human subjects were then assembled and forced to consume these tainted foods and beverages. Many became critically ill, with some succumbing to the cold hands of death. General Ishii discovered that the most effective conduits for these diseases were vegetables and fruits injected with the deadly pathogens. 7. Snake venom and hydrogen cyanide test. One day, A group of doctors from Unit 731 went together with an interpreter to a smaller unit at Nanking. Many human subjects were brought into a special research room. The interpreter lied to the subjects that these specialist doctors were there to treat them and give them medications that would do wonders for their health. Who would doubt a group of doctors smartly dressed in white coats, with shiny stethoscopes hanging around their necks? The poor victims trusted them and willingly offered their bodies for the supposed treatment. The doctors swiftly filled syringes with various deadly snake venoms and hydrogen cyanide, a highly toxic compound. They injected these poisons into the bodies of their victims. Then, sat down with pen and paper and meticulously observed the agonies endured by these beautiful souls. Within seconds, those injected with hydrogen cyanide began experiencing shortness of breath, followed by violent convulsions. They fell from chairs to the ground, losing consciousness. Others screamed, their eyes bloodshot with rage from the excruciating pain. All those injected with hydrogen cyanide died, and many snake venom victims too died. Next, epidemic hemorrhagic fever (EHF). Ticks were removed from the bodies of rats that were infected with EHF. About 200 of these ticks were ground and mixed with saline solution. This horrible mixture was then injected into the bodies of human subjects. The victims were studied and observed for symptoms of the disease. If the disease symptoms manifested in the subjects, his or her blood would be drawn and injected into another subject. The second subject would then be closely observed for the development of the symptoms. When they appear, the second subjects would be dissected, their organs removed, and part of these would be ground fine. Then a saline solution of the organ extract would be injected into another subject, and that person would be observed for symptoms. This process was repeated continuously until the pathogen was successfully isolated. 9. Testing toxic gases in pillboxes. In April 1942, when World War II was raging, Units 731 and 516 held a joint task to find the most effective method of spreading phosgene gas. This deadly chemical was used during World War I, and it caused thousands of deaths. This test murdered approximately 100 human subjects and lasted 3 days. 4 pillboxes were used for the experiments. 2 to 3 subjects were tied to a stake in each pillbox, and canisters of liquefied phosgene gas were thrown into the pillboxes. The victims quickly began coughing and experiencing chest tightness and difficulty breathing. As the gas continued to affect them, their symptoms worsened to severe wheezing and intense coughing. Fluid began to accumulate in their lungs. Breathing was difficult, and they felt as if they were drowning in a vast ocean. They kicked both hands and feet in a desperate attempt to save themselves. But soon, their kicking slowed as they drowned on dry land. The struggle faded, and they succumbed to the gas, unable to breathe any longer. Any subject that survived the initial experiment was put through once again. A 67-year-old man survived the experiments multiple times and other previous plague experiments. An army doctor injected air into his body with an unusually large syringe, and yet the man still survived. Hell-bent on terminating his life, this monster hung the man on a tree until he died. These guys had plenty of cages filled with rats, guinea pigs, monkeys, and other animals, but they chose to use humans. These scientists must have been trained in hell by the devil himself. Thanks for watching.